Hello everyone, my name is Haley Nate, and this is my presentation entitled Characterization of Halophyte Rhizosphere Microbiomes at Great Salt Lake, Utah. This was completed with the mentorship of Dr. David L. Parrott and the Westminster McNair Scholars Program. For those who may not know, the Great Salt Lake is located in northern Utah, just west of Salt Lake City. It is the largest saltwater lake in the Western Hemisphere, and it is the 8th largest terminal lake in the world. Because the Great Salt Lake is a terminal lake, no water flows out of it, so minerals like salt build up, making it extremely salty. For reference, the ocean has 3.5% salinity, while the south arm of the lake has 5%, and the reddish-colored north arm has up to 27%. But the salt doesn't just stay in the body of water. The water's edge has around 20% salinity, while the playa just above the water's edge has 5%, and distances up to 50 meters away from the water can have around 1%. Now, while salt is a natural part of life here, these extremely salty conditions can make it extremely difficult for many organisms to survive and thrive. This makes an extreme environment like the Great Salt Lake a unique host for a wildly diverse ecosystem of migratory birds, brine shrimp, as well as specialized plants and microorganisms adapted for high saline environments, such as halophytes and halophiles. Halophytes are commonly known as salt-loving plants. These are the specialist plants that have either evolved mechanisms to thrive in saline conditions, or they grow with the help of soil microbes. Some notable species of halophytes around the Great Salt Lake include inkweed, saltgrass, and pickleweed. Pickleweed, scientifically known as Salicornia, is a genus of succulent plant that is known for retaining water. This plant is most common around the edges of the playa nearest the edge of water where salinity is higher and there is less competition for growth. The roots of pickleweed function like the roots of any other plants, as they reside in a layer of soil called the rhizosphere. The rhizosphere of halophytes and non-halophytes is the same. It is simply the soil surrounding the root system of a plant. This region of soil is unique because it is directly influenced by any growth or nutrient exchange completed by the root system. Soil and roots are not all that exist in the rhizosphere, though. This is also home to the microbiome. The microbiome in and around the Great Salt Lake consists of specialized microbes known as halophiles, or salt-loving microbes. Halophiles mostly belong to the domain Archaea, but can also include the domains of bacteria and eukarya, a couple of common halophilic eukaryotes being algae and fungi. Now, within this photo of the Great Salt Lake, one may notice the difference in color between the southern and northern arms. The microbiome in the less saline south arm of the lake is filled with salt-loving halophiles, including green algae, which gives this end of the lake a greenish-blue color. The highly saline north arm owes its reddish color to the resident halophiles, mostly consisting of Archaea and red algae. These halophiles in the north arm contain carotenoids within their cells, just like in carrots, which makes them appear as various shades of red, pink, and orange, which you can see in this photo of the shoreline. Now, when billions of these halophiles grow together, they change the entire color of the north arm, which just goes to show that there is power in numbers. Now, in the surrounding soil outside of the Great Salt Lake, there is more of a mystery to the microbiome and how this microbiome may or may not be interacting with halophytes. And while the biodiversity of the salt-living plants around the Great Salt Lake is well known, what is not well known is the microbiome that lives in the rhizosphere around these plants, which leads us to the big question of what microbes are present in the soils around plant roots at the Great Salt Lake. This is significant because identifying the microbiome in the rhizosphere will be beneficial in answering future questions regarding how the microbial plant relationships function. To thoroughly characterize the microbiome within the rhizosphere of halophytes, we begin by collecting samples. We then use those samples to grow microbes, determining if they are present and to visually identify them, And finally, we use DNA sequencing to genetically identify the microbes. Samples were collected from 2 locations, one set from the less saline south arm of the lake at Antelope Island, and the other set from the more saline north arm of the lake near an art installment called Spiral Jetty, which was created by Robert Smithson in 1970. Our aim is to collect samples near the rhizosphere or root system of plants and next to the rhizosphere of plants to have a more comprehensive idea of where the microbes are living. For microbial identification, we grow microbes using specific types of media. Liquid tryptic soy broth, or TSB, and solid tryptic soy agar, or TSA, are used as generic growth media because the tryptic soy provides nutrients, which allows ample growth of most microbial species. The liquid and solid 18% saline modified growth medium, or MGM, is used as a selective media for salt-loving halophiles. We begin the growth process by placing a small amount of a soil sample into 4 tubes. 2 tubes will have liquid TSB media, and each will be placed into these shaking incubators, one set to 30 degrees Celsius and the other set to 37 degrees Celsius. The remaining 2 tubes will contain the 18% saline MGM liquid media, each tube placed into the same 30 and 37 degrees Celsius shaking incubators. Now, once growth occurs in liquid media, we can then inoculate or streak the growth onto a plate containing the same media and incubate it at the same temperatures. The plates are streaked like this in 4 main quadrants, which allows isolated colonies of microbes to grow so we can better view them under microscopes with the aid of simple staining and gram staining techniques. Simple stains include only one dye, such as methylene blue, which will help show the outline of microbial cells, making them easier to see when using a microscope. Gram staining is a technique that uses more than one type of chemical to stain cells, helping us differentiate between bacteria that do or do not have peptidoglycan, which is just a special layer of sugars and amino acids in the cell walls of some bacteria species. Gram-positive results will appear purple, signifying the presence of the peptidoglycan layer, while gram-negative results will appear reddish-pink, signifying the absence of the peptidoglycan layer. In addition to physically identifying the microbes within a sample, we also have their DNA sequenced to identify individual species of microbes. To do so, we begin by using a kit that extracts microbial DNA directly from soil samples. We then run 3 different 16S rDNA PCRs on the sample, using specific primers for eukaryotes, bacteria, and archaea. PCRs, or polymerase chain reactions, are used to amplify segments of DNA by making billions of copies of the extracted DNA. 16S rDNA just means this is a broad-range amplification, which will help us in identifying more sequences of DNA for a species in the microbiome. With completed PCR samples, we then need to determine which samples contain the highest concentration of DNA. To do so, we use a technique called gel electrophoresis, which utilizes electric currents to move amplified DNA from the PCR samples across the gel, which you can see in this picture. Smaller fragments move farther across the gel than larger fragments, and an indication of successful sample is the appearance of a band. The brighter the band, the more DNA present in the sample, making identification easier and more accurate. Now, having identified which samples contain the most DNA, the successful PCR samples are mailed to a genetic sequencing company called Mr. DNA for 16S rDNA sequencing and sequence analysis. Preliminary data representing rhizosphere soil samples was provided by Mr. DNA and analyzed by now graduated Westminster students. This graph compares the differences between microbiome composition of Archaea found at the 2 testing locations around the Great Salt Lake. Taxonomic Archaea orders are listed along the x-axis, and the composition percentages of each order are listed along the y-axis. The composition represents the percentage of orders found in each location. The blue bars represent the less saline south arm of the lake at Antelope Island, and the orange bars represent the more saline north arm of the lake at Spiral Jetty. Our preliminary data showed 58 Archaea orders in total. The presence of more blue bars compared to orange across the x-axis signifies that Antelope Island hosts greater biodiversity than Spiral Jetty, and the height of the orange bars signifies that Spiral Jetty hosts a greater percentage of certain Archaea orders compared to the same orders from Antelope Island, the most notable order being Halobacterialis, with a composition that was 39% higher around the Spiral Jetty than it was around Antelope Island. This graph compares the differences between microbiome composition of bacteria found at the same two testing locations, represented by the same colors as the previous graph. These taxonomic bacteria orders are listed along the x-axis, and the composition percentages of each order are listed along the y-axis. Further preliminary data analysis of rhizosphere soil samples showed 1,100 bacteria orders in total. Similar to the previous graph, biodiversity of bacteria orders was greater at Antelope Island than at Spiral Jetty, and Spiral Jetty had a greater percentage of certain bacteria orders compared to Antelope Island. The two most notable bacteria orders being Flavobacterialis, with a composition that was about 28% higher at Spiral Jetty than at Antelope Island, and the order Bacillialis, with a composition that was about 33% higher at Spiral Jetty than at Antelope Island. Based on these preliminary results, we expect future results to reflect a greater biodiversity around the less saline south arm of the lake and a larger number of halophilic orders around the north arm of the lake. Now, having completed successful DNA sequencing processes, we have gathered enough information to at least know there is evidence of microorganisms living in the rhizospheres of halophytes, which leads us one step closer to understanding halophile-halophyte relationships around the Great Salt Lake. And, based on our preliminary data, it does suggest that between the north and south arms of the lake, there is a difference in biodiversity. What our data doesn't explicitly confirm is if the difference in salinity between the north and south arms of the lake is a factor in biodiversity. Future research is currently underway as we are collecting more samples from Antelope Island and Spiral Jetty locations. We will also be collecting from some new locations at the north and south arms of the lake that have yet to be determined. Along with collecting new samples, we are also researching ways to determine the salinity of the soil to better conclude if salinity is a factor in biodiversity. And once we have more samples that have been successfully sequenced, we plan to use qPCR to quantify the number of microorganisms present within the samples. And lastly, we hope to expand a budding community outreach program, which uses uniquely colored microorganisms found around the Great Salt Lake to create art using agar plates like this one of a pelican. The goals of this outreach program is to help others learn about the microbiome around the Great Salt Lake like some of you have today. Here are my references, and I would like to take a quick moment to thank my mentor Dave, the Westminster McNair staff and cohort, and all of you for taking the time to watch my presentation. If you have any questions for me, please feel free to scan the QR code to email me directly and stay connected. Thank you.